Thank you, Huanyu, for your very nice introduction. Good afternoon, everyone. Firstly, I would like to take this chance to thank the organizers, in particular Professor Tsinghua and Professor Yuxin Zhang, for their kind invitation. I really appreciate this opportunity to introduce the research of my group. So today I'm going to talk about a recent work of my group on the development of bio-inspired electronic skin. Well, we know that most biological systems and human organs have very complex while delicate 3D construction at the micro and the nanoscale, such as the double helical nanostructure of DNA, the spatially folded protein structure, ellipsoid-shaped mitochondrion, and tree-shaped interneuron. These micro and nanostructures not only form naturally in biology, but also provide the most essential functions. As an example, the hierarchical 3D surface morphologies of many insects and animals are very crucial to their unique functionalities. For instance, we know that the gecko can swiftly travel across the walls thanks to the nanostructures of their gecko tool, which allow them to tune their adhesion with the walls very swiftly. The microstructure of the locust leaf enables its superhydrophobicity such that the water cannot stay on the leaf. And also the shark skin consists of well-aligned 3D microstructures, which is very crucial to the drug reduction during their swimming. Actually, these micro and nanostructures are not far from our life. The electronic devices that are built from architected micro and nanostructures just correspond a very good example. Actually, for quite a long time, since the 1960s, the development of integrated electronics follows the prediction of Moore's law very well. However, during the last 10 years, we can also see an evident lag between the development of integrated electronics and the prediction of Moore's law. This is mainly because the planar microfabrication techniques have already approached their physical limits. So how to continue the Moore's law? We need to introduce new ideas and new rules for their further development. Considering the third dimension can bring brand new opportunities, the 3D microelectronic devices have already become a very important research direction in the post-Moore era. So what's the advantages? In general, when comparing to 2D designs, the 3D electronics can provide broader design spaces, better mechanical properties, improved device performances, and even brand new functions. For example, Professor John Rogers' group from Northwestern University has made a very inspiring work on the bio-inspired electronic eyes. So in this specific work, inspired by the unique structure and functional features of compound eyes, they have developed an artificial vision system capable of high-resolution imaging with an ultra-wide field of view and also an infinite depth of field. So both of these two features are unachievable based on planar 2D device architecture. So just because of these outstanding advantages, the 3D electronics have very important applications in many areas, such as bio-integrated electronics, microrobots, and healthcare, with the potential to upgrade their device forms. The electronic skin is just one of such areas with both fundamental and practical significance. On one hand, by mimicking the tactile sensing capabilities of human skin in static force, strain, temperature, vibration, and etc., the electronic skin can be integrated with robotics to achieve robotic sensing and to improve human-machine interaction. On the other hand, the electronic skin can be also integrated with our own human skin to serve as a second skin for long-term monitoring of biomedical singulars, such as blood pressure, core body temperature, blood glucose, blood oxygen, and so on. So many inspiring progress has been made in this exciting area of electronic skin, and this slide summarizes some of the milestone works published in Nature and Science from 2011 to 2024. So those works on the top mainly fall into the type of inorganic electronic skins, and those on the bottom have some key electronic components made of organic electronic materials. So since the seminal work of abdominal electronics done by Professor Zhang Raja's group in 2011, there have been many significant advancements along the path of inorganic electronic schemes, such as the biloadal wireless electronic system for neonatal intensive care, the wireless haptic interfaces for VR and AR, and highly conductive and elastic nanomembranes for skin integrated systems. If you look at the pioneering works on the organic e-skins, you will see many of the pioneering works were done by Professor Zhang Bao's group from Stanford and also from Takosu Mayo's group from Tokyo. Although there have been many inspiring progress in this field, two grand challenges still stand out. So challenge one is decoupled sensing of multiple static loads related to tactile sensing. So in general, there are three different types of mechanical parameters related to tactile sensation. For example, when you touch an object, there will be normal force induced by the touch, which correspond to the pressure. There will also be shear force, which correspond to the frictional force, and also it will induce the skin deformation. So when all of those three different parameters come into play, it becomes very difficult for the device to discriminate and to measure simultaneously. Actually, Professor Huang Yuchen's group wrote a very nice review paper on this topic of multimodal sensor with decoupled sensing 3 years ago in Advanced Science. And so this is challenge one. Challenge two is the quantitative measurements of multiple physical properties. For example, if you want to use the e-skin to determine the softness of an object without knowing its shape, it means that you need to determine both the elastic modulus and the principal curvature components simultaneously. This is also very difficult to achieve. So to address these challenges, we look into our own human skin to find inspiration, and we propose a bio-inspired 3D layout of sensing components to achieve decoupled sensing of those mechanical parameters. The standpoint is to leverage the biomimetic physical architecture of the device to achieve decoupled sensing. I will also introduce a mechanics-guided 3D assembly approach to allow the fabrication of the 3D e-skin. Of course, this e-skin device can be integrated with the data acquisition and the signal processing modulus to form a tactile system for simultaneous measurements of elastic and local shape. Well, I will first introduce the bio-inspired design of the 3D e-skin. In our human skin, there are two main types of slowly adapting mechanoreceptors, namely the Merkel cells and Ruffili endings. The Merkel cells are located very close to the skin surface in the epidermis layer, while the Ruffili endings are deeper in the dermis layer. Because the force applied to the skin decays rapidly along the depths, the Merkel cells becomes very sensitive to the external forces, while the Ruffili endings deeper in the skin are also sensitive to the external forces. Additionally, we notice that the Merkel cells are supported by collagen fibers in the dermis layer, which makes this part to be rigid, more rigid than the other part. Therefore, when you apply a stretch to the skin, the soft part will undergo a larger deformation than the other region, and therefore, the Ruffili endings become more sensitive to the deformation of the skin, such as the stretch of the skin. So this is the mechanism why the Merkel cells and Ruffili endings in our human skin are more sensitive to the external forces and the stretch of the skin, respectively. So to mimic such human mechanosensation, we propose a bio-inspired design concept of the 3D e-skin device, which also consists of 3 different layers, including the epidermis, dermis, and the hypodermis layers. And more importantly, the force and strain sensing components are distributed in a 3D layout, which mimics those of Merkel cells and Ruffili endings in our human skin. So you can see the force sensors are very close to the skin surface, and the strain sensors are deeper in the dermis layer. And also in this work, we propose to use the piezoresistive sensors to mimic the slowly adapting receptors in our human skin, because they can produce continuous outputs in response to sustained streamline. And then the signals generated from those sensors can be collected and processed through the modules of data acquisition and signal processing, which is similar to the function of central nervous system in our human body. So to this end, I have introduced the bioinspire design concept of the 3D e-skin, but how to fabricate it, especially noticing that the human mechanoreceptors in our human skin have a very high density distribution. And the spacing between those mechanoreceptors just range from around 100 micron to several hundreds of micron. So to allow its fabrication, we exploited the mechanics-guided 3D assembly approach, which was initially developed by my group and groups of Professor John Rogers and Professor Yingguang Huang. The key concept of this 3D assembly approach is to go one step further beyond the traditional 2D fabrication, and we introduce controlled compressive buckling to drive the transformation of easily patterned 2D structures into well-controlled 3D architectures. Because the planar microfabrication can do mass production, which means thousands of components can be fabricated at one batch. Our 3D assembly approach is fully compatible with the 2D fabrication techniques, so it can also assemble thousands of 3D metal structures and even devices at one time. So because of the complex mechanical loadings that can be applied and the compatibility with the 2D fabrication approaches, this 3D assembly approach has many unique advantages in terms of the applicable material types, length scales, and diversity of 3D geometrical shapes that can be formed. So this approach not only applies well to inorganic semiconductors like single crystal silicon and gallium nitride, it also applies very well to metals, polymers, plastics, ceramics, and even piezoelectric materials like PVDF and PZT. And also, unlike many of the other fabrication approaches, this assembly approach can work very well over a wide range of length scales, from some micron scale and nanoscale to even meter scale. So if you are interested in this 3D assembly approach, you are recommended to read our review papers published in recent years. And then I will introduce the use of this 3D assembly approach to fabricate the 3D e-skin. If you look at the image on the right, you will see an expanded view of this 3D e-skin device. So this device consists of 3 different parts, including the functional part, the substrate, and the encapsulation. This functional part is initially flat and has a 9-layer construction, including 2 force sensing layers, 2 strain sensing layers, and 5 polyimide layers that encase each of those sensing layers. And the video on the left shows the 3D assembly process of this device from initially 2D format into 3D construction. And after the 3D assembly, we introduce a heterogeneous encapsulation strategy to fill the inner regions of those cage metal structures with relatively rigid polymer and the other regions with relatively soft material. And this is mainly for the purpose of strain isolation. Okay, let's look at the optical images of the fabricated 3D skin sample and the magnified views of 2 functional units, force sensor, strain sensor, and the via. So you can see this cage metal structure has 8 arms, and on top of each arm, we arranged a force sensor, and the height is around 600 micron. In comparison, the strain sensors are located on top of this arc metal structure with a low height of 250 micron. So this height difference can ensure the decoupled sensing of the force and the strain. And additionally, the distance between the center of this cage metal structure and this one is 2.5 millimeter. So the entire device with 5x5 array has a comparable dimension to our fingertip. And over this one fingertip region, we have 204 sensors and 40 strain sensors in total, which is close to the density of mechanoreceptors in our human skin. Great. Then let's consider a functional unit of this device under combined loadings of normal force and shear forces. And because of the geometric feature of this cage metal structure, we can correlate the normal force and 2 shear force components explicitly with the relative resistance changes of these 8 piezoresistive sensors. This means that this kind of 3D cage configuration can ensure that both the normal force and shear force components can be measured simultaneously. And then I will use mechanics simulations to illustrate the decoupled sensing mechanism of the force and the strain. So if you look at the video here, we can apply a pure pressure loading to this device. And then you can notice that the deformations induced in these strain sensors are much smaller than those in force sensors because those force sensors are very close to the skin surface. Similarly, under unilateral stretching, the deformations in those force sensors are negligible because of the strain isolation of the cage method structure. So you can see almost zero electrical outputs from those force sensors. You can only see output from those strain sensors. And also, if you apply a pure shear loading to this device, the deformations in the strain sensors are also negligible. And therefore, a fully decoupled sensing of normal force, shear force, and strain can be achieved. Okay, then let's look at a set of experimental results. We firstly apply a pressure to this device, and this red curve denotes the pressure response. And then this is shear force response, green color. And then we apply a unilateral stretching to this device, and you can see the strain response in this blue color. And by holding this unilateral stretching, we can apply an additional normal force and an additional shear force. So this is in real time, and this set of experimental results validate the decoupled sensing capability of this 3D skin device. Then I will illustrate the characterization of the sensing performance. So if you look at the mid-top image, it shows the sensing performance of pressure with a linear range of 80 kilopascal and a response time of 250 milliseconds. And after 3 sovereigns pressure loading and unloading, the pressure sensing performance is still very stable. If you look at the results here, it shows that we apply the shear loading to the device along 6 different directions, and the predicted shear force magnitude and angle agree very well with the prescribed values. And in terms of the strain sensing, we stretch the device and unload up to 10,000 cycles with a strain amplitude of 20%. And after the fatigue loading, the device is still performing very stably. Great. Then this 3D e-skin device can be integrated with the modules of single acquisition and single processing to form a tactile system. Previously, there is no tool that can be used to characterize the rich information of tactile sensation. So in this work, we propose a spatial temporal map to visualize the rich mechanical information of tactile sensation in real time. So you can see there are 25 octagons in this spatial temporal map, which corresponds to the 25 functional units in the device. And the color or the size of each octagon denotes the magnitude of the pressure. And the black arrow denotes the magnitude and direction of the shear force. And also the spacing, horizontal spacing and vertical spacing between neighboring octagons denotes the normal strain components along the horizontal direction and the vertical direction respectively. And then we can take a look at this video. We use this 3D e-skin device to touch 3 surface relief structures of letters T, H, and U. We first use the device to touch the letter T, and you can see clearly the symbol from the spatial temporal map. And then we use the device to touch the letter H, erasure to the side of the letter, and then we move the device to the center of the letter. And during the touch, we also apply a friction force, so you can see this information from the spatial temporal map. And finally, we use the device to touch the letter U, and during touch, we also apply a torque. So from this kind of spatial temporal map, you can identify which kind of mechanical loading is applied to the object. This is also the first time we can visualize the rich mechanical information of tactile sensation. For our human body, when we touch an object, we can use the induced force distribution and the skin deformation to infer the softness and the local shape of the object. Similarly, our tactile system can also distinguish the softness and the local curvature components of two different objects. So based on this principle, we can develop a deep learning neural network model to correlate the elastic modulus and principal curvature components of the object with the relative resistance changes of 240 sensors in our device. So to do the machine learning, we need to construct a large data set. So in this work, we constructed the data set by touching a diversity of different samples with 50 different surface shapes and 15 different elastic moduli. So in total, the data consists of 30,000 samples. And this figure shows the testing results of 2,700 samples from the testing set. So from this set of results, you can see good predictions of the elastic moduli in the range from 40 kilopascal to around 4 megapascal. And also very good agreements can be observed between the prediction of principal curvature components and the true value. So this kind of 3D skin device can be integrated with robotics for many different applications. So in this example, we use the device to measure the softness of 4 different fruits, including the peach, kiwi, plum, and plum, in both unripe and ripe conditions. We have also used the device to measure the elastic modulus of 4 fermented bread and cake in both the fresh and dried states. We know that the fruits become softer as they ripen, but the bread hardens during their air exposure. But previously, we don't have a tool to quantify the value of the softness. So based on our developed system, we can use the eSkin device to measure quantitatively the elastic modulus of the fruit in both unripe and ripe conditions. Just within 1 second, we can determine the value of the elastic modulus that characterizes the softness. Okay, so in summary, I have introduced a 3D architected electronic skin device that mimics the human mechanosensation. I hope I have convinced you that the BioInspire 3D layout of sensing components provides an effective route to fully decouple sensing of normal force, shear force, and strain related to the tactile sensation. In a very recent perspective paper published in ACS Nano, we have summarized the representative 3D microstructures that have been exploited in tactile e-skins, including the cages, frustums, pyramids, pillars, domes, lattices, and so on. Actually, each class of those 3D microstructures offer unique advantages in capturing certain types of tactile singulars as marked by those collections. And the right part of this fig links a collection of physical properties and surface features of the object with specific type of tactile information required for their prediction. So if you are interested in this part, you are recommended to read our recent paper. And excluding the electronic skin, there are also many other opportunities in the exciting area of BioInspire 3D electronics because of the gaps in the dimensions, structures, mechanisms, functions, and intelligence between the natural organisms and artificial electronic systems. So there are rich opportunities in the design and fabrication of 3D architected electronics and systems in many different aspects. And here I just list some interesting directions, and all of you are very welcome to explore together along this direction. And finally, I would like to thank my collaborators and all the current and former members of my research group. It is them who did all of those works, and I just introduced those works in representative of them. And also, I want to thank the funding support from different agencies. And with that, I would like to thank you for your attention.